All right. So what we're going to do in this class is finish up with biosynthesis and then look at molecular biology. I think the molecular biology topics are really exciting, and I like how it's done in this class. What you're going to do is talk about the theories of how things work. And a lot of times textbooks give you the picture of how this all works together, how initiation happens, how translation happens, things of that nature. But what we were going to do in this class is do a little bit differently. You might have a little picture that gives you an overview, but what we're going to do is be presented with the actual experimental data that actually caused the PIs to come up with the theory. And so it isn't a cartoon look, it's a real look at real data, and it's a lot of blotchy little blocks with dark spots or light spots or kind of more darkish, more lightish to compare different experimental conditions. And this is where we want you to get. We want you to get to a place where you look at the different experimental conditions that were set up, you look at the outcome, the results, the data, and from that, then you build a model that you can describe for what is actually happening. And so a lot of the models that you've seen already, like in genetics, you've seen the cartoonish and you've described it and you understand it, but now you're going to see how we came about getting to those models based upon data. And I think this is really interpretive and one of the culminating places we want you to get to, because past this, if you're learning anything, it's going to be from those experimental conditions and real journal articles, which may change the picture that we teach you. In general chemistry, everything that we teach you doesn't change. Okay, you only have so many weeks. What we can cover is covered. It's been pretty much, we know what's going on. Organic chemistry the same way. Even though there is research in organic chemistry, what changes doesn't happen in between the first week of the Organic 1 and the last week of Organic 2. All of the new things happen in graduate school, and so textbooks are often wrong. Biochemistry and molecular biology are the places where we get to in which the textbook version that you have may or may not be the current idea of what's happening because more research has come out, and so it's changeable. For us in the sciences, sometimes that's uncomfortable. We like to know what we know. We like to know that we get it right. But at the point at which we are now, there are different groups that are competing about the ideas of how things happen based upon data, and so you're going to have to get to that point of being able to justify what the author says by the data that they present. And so that's a different step, but it's a learning step, and that's why we're now in a 400 level class. When we looked at metabolism, okay, what were the main types of pathways that we looked at? Where did we start? Glycolysis, because we are intending to break down what? Glucose. And glucose is what we think of as energy, right? But that's not all the energy, correct? What other things are energetic? Fats and proteins. And so what we did was we broke them all down individually, and what we did was we oxidized them to CO2, to water, for the carbons and hydrogens. We extracted the electrons, we created a proton gradient, and that proton gradient then created ATP. All right? And so that's part of metabolism, is breaking things down. Well, if anything is broken down, what can we do? We can make it. And so we're going to look at synthesis of those three categories, fats, proteins, and the sugars themselves, although we actually looked at sugars in photosynthesis. We're also going to look at synthesis of the nucleotides, because those are kind of important, right? The nucleotides, and we're going to look a little bit at both synthesis and degradation. Then what we're going to do is we're going to integrate all of metabolism. And I've thought about that chapter and what we're going to do for your poster session that you're required to do this year. We're going to change it from what we did in the past. What we're going to do is we're going to ask you to create posters in groups, but what I want you to do is do something in real integration of metabolism. I want you to take a diet. You know, one of those diets that you could try, a keto diet, a paleo diet, a whatever diet, and I want you to, and we'll give you more details, but in general, I want you to think about that diet and see how does it change your metabolism? What are the pathways? What are the main sources of energy? How does it integrate? Okay, and so we will break up into groups, assign groups, and then pick different types of dieting fads that are out there and see what that might do to metabolism. And I think that's a great way to see, do you understand how everything works? Also, it may surprise you about how some of these diets work. And you may not find exactly anybody has that kind of information, because a lot of those diets are made up by people that have no idea about the chemistry or the biochemistry that happens behind it. They just say, take this, the juice pickle diet, you know, and what is it going to do? Well, yeah, you might not take in any calories. Oh, you lose weight, that's great, but what does that make your body do and your metabolism? So we'll just giving you a preview of what that's going to be in the future. All right, so what we're going to do is we're going to start to synthesize anything that is hydrophobic, or the fats. That'll include the fatty acids, that'll include the membrane lipids, and that will also include cholesterol and its derivatives. And so if we are going to look at fats in synthesis, well, if we remember way back when we broke down fats, when we broke them individually down, we created acetyl-CoA. And so, being acetyl-CoA, this is a big branch port. We're going to start with acetyl-CoA and do synthesis. But acetyl-CoA is really kind of the degradative portion of it, so we need to make it go down to the synthetic portion, and the conversion to malonyl-CoA, right here, is the committed step. All right, and so we could take acetyl-CoA and break it down to CO2, or we could add to it to make malonyl-CoA. So what do we add to this to make it malonyl-CoA? Okay, the ion is what? Bicarbonate. Okay, that's great. But bicarbonate is, what do we think dissolved in water? CO2, carbon dioxide. All right, and so if you add a carbon dioxide to a organic structure, what you're going to end up with is an acid. But we saw that before. A lot of CO2 was produced from decarboxylation of an acid, so it's the reversed reaction. The first and committed step is done by 3 active sites, okay, 2 active sites with 3 proteins. In the prokaryotics, and in the eukaryotics there is 1 protein with all of these domains. All right, the first thing that we are going to do is we are going to synthesize the activated species. The bicarbonate reacts with this unit called biotin. It's also called vitamin H. You don't hear about vitamin H too often, but that's what it is. And the CO2 is temporarily placed onto the nitrogen of the biotin. Okay, and so this biotin carrier domain has this long lysine, and here is the vitamin B, and you see this long tail, and you see the ring structure. We add the CO2 onto that, okay, and it will transfer the CO2 into the active site back and forth when we do the reaction. Now, the reaction that we ultimately want to do is we want to have this CO2 carbon here to make malonyl-CoA. All right, and so what we're going to do is this CO2 here has a wonderful, okay, this biotin is a leaving group. And so I want to add this carbon to that carbonyl. Now, what do we know about this carbon that is sitting in the acetyl-CoA? It is next to what? That methyl group sits right next to a, what's that functional group? Carbonyl, right? What does that make these protons? They're slightly acidic, okay? And so what we can see happens, if I articulate all of the hydrogens, the methyl group is slightly acidic, and if I do my inventory, carbonyl, carbon, 3 hydrogens, carbonyl, carbon, 2 hydrogens, that tells us we're going to have to get rid of one of those. So I take one. This negative charge then attacks at the carbonyl. I can kick up, I can kick down, and I have the addition of the acid to get to malonyl-CoA. Malonyl-CoA is destined for synthesis. Now, one thing about the carriers, we talk about acetyl-CoA all the time. Okay, acetyl-CoA, well, the CoA portion is this. Okay, it's huge. It's this very long, from the sulfur, very long organic compound, and then we got 2 phosphates, we got a ribose, we've got an adenine, and so this is pretty large. Here's the acetyl group sitting right there, okay, on that sulfur. Well, if things are on the acetyl-CoA, that tends to take them towards degradative pathways. We need to really get this molecule into the synthetic pathways. Another carrier that can be used is a small protein. It's called acyl carrier protein. And what it does is it has a small molecular weight, and it also has that same organic structure as acetyl-CoA with the same sulfur. In terms of whatever is attached to this sulfur, it really doesn't matter organically. It looks the same. So from the perspective of this group or the perspective of this group, whether it's attached to acetyl-CoA or whether it's attached to the ACP protein, it can't tell organically. But this part of the molecule tends to take it to the degradative enzymes, and this protein tends to take them to the synthetic proteins. So that's what the difference is. 4 reactions to do synthesis. Well, let's do a review. What are the 4 reactions to do degradation that you learned last semester? Oxidation, hydration, oxidation, thiolysis, okay? In reverse, okay, thiolysis is breaking. I condense. Oxidation, the reverse, is a reduction. Hydration, the reverse is dehydration. And oxidation reversed is a reduction. We do the 4 steps in reverse, okay? And so they are related. So the acetyl-CoA comes in first, and it reacts with something called Ks. Well, Ks is the keto-acyl ACP synthase, okay? The acetyl group gets transferred onto the protein. Okay, that's important. The second thing, the malonyl-CoA that comes in, okay, malonyl-CoA gets converted to malonyl ACP. That means it's going to be transferred into the synthetic portion. Okay, so I've got an acetyl-CoA primer, I've got the activated species, and what I'm going to do is do a synthesis where I start off with 1, 2 carbons. I bring in 1, 2, 3 carbons, and I make a 1, 2, 3, 4 carbon intermediate, and I lose CO2. So 5 carbons up here, 4 in the growing fat, and then this is a little tiny growing fat chain, and then CO2 is lost. All right. Just like we did degradation, we started with evens, we took off 2, we took off 2, we took off 2, we took off 2. Here we're going to add 2, add 2, add 2. The reason why we put CO2 onto the molecule to make it malonyl-CoA is to really activate the reaction. Now, activating the reaction has been up to this point almost always using ATP. We have a good leaving group. Phosphate is a wonderful leaving group. All right. CO2 is also a wonderful leaving group. Even better than phosphate because it can bubble out of solution. And so, let's do some inventory, okay? This is, let's start 1, 2, 3, 4, 5, and up here we'll go A and B. All right, so down, if I do this, that's 1, that's 2, that's 3, this is 4, okay? And if I do this, this is B, and that is A. So what has to happen is, that must be 5. All right. What we have to connect is which carbon number to what carbon letter? What needs to be connected? 4 to A. Okay. Well, what do we know about A? What charge does A have partially? It's slightly what? Positive, because the electronegativity difference. So I need to get something negative. So I need to make 4 a little bit negative. Well, how can I do that? If I take, and let me articulate all the lone pairs here. It's never written, but they're always there. Okay. To make CO2, I'm going to take one of those lone pairs, and I'm going to form a double bond. Okay. I get the Lewis structure that looks just like this, which I assure you I expect a GChem student to be able to draw. All right. And what happens is there's this pair of electrons that is right here. This pair of electrons can sit on this carbon for just a moment. What could that do? Well, it could resonate, there's stability, but it can also act as a nucleophile, and we've seen this before. So what I'm going to show is the electron pair attacking. Every time I attack something that has a double bond, what has to happen? Kick up, kick down, and then something has to leave. In this case, it's going to be the enzyme itself. And now we have the growing chain on the ACP. So that was the condensing. I made a carbon-carbon bond. Our next reaction is a reduction, and it's done by an enzyme called a reductase. It uses an electron carrier, NADPH. What do we know about NADPH? Where was it made? Where do you make all your NADPH? If you forgot this quiz, I'm going to assign it again. The pentose phosphate pathway. We make lots of NADPHs there, right? NADPH is a little bit different than NAD plus and FAD in what regards? What happens is they're all 3 electron carriers, NAD plus, FAD, and NADPH. They will carry electrons. 2 accept, generally, and 1 donates. The NADPH is the donator. It's the one used in synthetic processes. And so this is going to donate a hydride. And obviously, it can react. The hydride's a nucleophile, and it could conceivably attack here, or it can attack right here. What does it do, blue or green? Blue. Why does it do blue? Other than the fact that you see in the product, that's where the change happens, right? But why does it do blue? Is it hindered? A little bit it is hindered, but that's not really the case. In an organic flask, if I put this in solution and I just put that in solution, I would have attacks at both, and because of sterics, I might say 60 to 40 if it were just sterics. But I would get both products. In the body, I get this product only. Why does it only attack at one place? Yes? Correct. Remember, it's always the enzyme. The enzyme holds the two substrates and places them in a position where the attack can only happen in one place. And so I will attack in the blue. I kick up. That's something that we've done all the time. And when you attack a carbonyl, you can kick up and you can kick down. Okay? Do I kick down in this particular reaction? No. How do I know that? Because there's no more carbonyl. So what is my other option? I can kick up and then pick up a proton. And you always have a proton available inside the enzyme. Remember, it's one of the amino acids in the active site. What is the third thing that could happen if you attack a carbonyl? It can kick up, kick down. It can kick up and pick up a proton, or it can kick up and do what? There's one more thing it can do. It is a negatively charged intermediate. It could attack something like ATP. It could act as a nucleophile. And we've seen once or twice that that happens, that it can attack itself something else. All right, so a hydride is represented by this hydrogen and this pair of electrons, and an acid reaction that's catalyzed represents this hydrogen. And so I have a hydroxide. Okay, and that's the reductase. Okay, we're going to dehydrate. All right. And in this case, what in the world, when we do inventory, what happens? Okay. What in the world would be our first reaction for this? Base. Because it's all about the, right? It really is. A lot of times, 90 some odd percent of the time, we're going to start off deprotonating something, okay? And so what we're going to do is we're going to deprotonate this proton, okay? And we will deprotonate that, and the actual side chain that is used for that is a glutamic acid. When that deprotonates, okay, then what are we going to do with the pair of electrons that were between the hydrogen and the carbon? That's going to form the what? The double bond. And then what will fall off? What's the leaving group? In this case, it will form water because I'm going to have a proton, and this pair of electrons will fall and pick up the proton to form the water. One of the very, very first reactions that you learned, usually in organic chemistry, is this elimination. Okay? Is this elimination E1 or E2? We have elimination. Okay. Is it an E1 mechanism that I drew, or is it an E2 mechanism that I drew? Okay. Discuss with your neighbor. Why in the world is he asking us about E1 and E2? I don't even know what E1 and E2 is. All right. We got it. We had enough time to look at each other, look cross-eyed, and okay, what am I going to guess? Okay. How many people says that your partner said it was E1? How many people said your partner said it was E1? Okay. How many people said your partner said it was E2? Okay. And then how many didn't vote? Yeah, there was a lot of different voting. Okay, so E1 eliminations. E1 eliminations have the leaving group leave first, a carbocation is formed, and then you deprotonate to fulfill the double bond. An E2 mechanism, okay, and why it's an E1, the rate is dependent upon the leaving group only, okay? and that's why it's E1. E2 has to have both things involved, the deprotonation and the leaving group, so the base and the reactant are part of the rate mechanism, and it happens in, I take the base, deprotonate, the electrons kick in, and then the leaving group leaves. Okay, knowing that, how would your partner vote now? Is this an E1 or an E2 mechanism? E2 mechanism, because there was no cation that was formed in this reaction. We will see some E1 eliminations later in this chapter. Okay, so if you need to know about an E1 and an E2, what else do you need to know in terms of organic mechanisms? SN1s and SN2s, we'll get to those in other reactions also. All right, our next reaction is done by a reductase. All right, and so I have a choice for you. Let's do green. Okay, watch, just watch. That is an option. And let's do not red for the colorblind people. Red and green are not good choices. All right. Which do you think happens? The blue or the red? I'm sorry, the blue or the green? They look pretty similar, don't they? Right? But there is an inherent intermediate that I didn't draw, but I want you to understand. Okay? The first line of either one happens, right? The second line that I draw is a two-step thing that happens. Okay? Let's do the two steps in green. That is an intermediate, and then I pick that up. The blue would be, that is an intermediate, and then it picks up. So inherent in this reaction, there is going to have a carbon that has a negative charge for just a moment in time. Which one is better having the negative charge? The green or the blue? Blue. Because the negative charge is on a carbon that is alpha-tocarbonyl. If by chance it's on there for a second, it can go and do its reaction, but it can also resonate, which lowers the overall energy. If I do green, that negative charge is on a carbon that is stabilized by nothing else. The enzyme uses internal stabilities of reactions of the actual organic mechanism to help it. All right, so that's a whole lot of just really, really nasty writing there. I'm going to go back and try to take off stuff to make it look better. Okay. I don't know how far back I have to go, especially when I do those little dot things. I got to take off every dot one at a time. Okay. Oh. Hey, I got it. Okay, so we will attack here with the electrons. Okay, and we will pick up a proton. All right. What have I done? In 4 easy steps, I have doubled the length of the little fat. Okay, it's a 4. All right, is 4 the normal fat? No. What is the normal length of a fat? 16. Okay, so we repeat the process. The activated species is the malonyl-CoA that comes in. Now, what's interesting about this, if you take a look at the at your notes, there are a lot of different active sites in this process, and you can see better in your notes than on the board right now. There are individual proteins that are bunched together. Okay, originally we looked at it in prokaryotic systems, the bacterial systems. We actually identified 7 separate proteins. If we look at yeast, that goes down to 2 proteins, and if we go to vertebrates, we're down to 1 single protein, although we do have 7 active sites, 7 functional pieces in this. And so the fat just gets moved around from place to place to place as each reaction happens, okay? And the ACP, the acyl carrier protein, will actually move the substrate into different active sites as it goes around. Only once does it get moved in one reaction to the KS enzyme, but we go round and round and round until we get to 16. What is a little bit more interesting in the vertebrates is that this bundle of active sites, that moving around actually happens between two pairs of subunits that have 7 active sites, and it moves back and forth. This actually happened, and we saw this with the sugar synthesis, that it moves, each time something gets longer, it moves back and forth between subunits. And so that seems to be a common strategy in synthetic processes, moving between two identical subunits of the functional groups. It will take 7 reactions to synthesize that fat, 7 ATP, 14 NADPHs to get this all done. We'll have 8 CoAs that are leaving groups and 14 NADPHs. This represents a very large amount of energy. Okay, 7 ATPs is pretty bad in and of itself in terms of energetics, but those NADPHs, for each one of those, or for every 2, you actually take glucose through a cycle and lose a CO2 to get that done, and so it uses a lot of energy. Well, fortunate for the American diet, this doesn't have to happen a lot. Okay, we intake enough fat that not a lot of synthesis has to happen. Okay, but we do have drugs to stop some of these synthetic processes from happening because some of these enzymes run even when they're signaled not to. Okay, special cases. Sometimes we have fats that are odd numbers. Instead of using the acetyl-CoA as the starting point, we'll use a propanil-CoA and then transfer it to the propanil ACP. So that can get us an odd fat. Okay. NADPH is used for all of the reductions. Normally we synthesize up to 16 and then we stop. If I need to elongate past 16, it goes in other compartments and done by another set of enzymes. Okay. Unsaturations are performed by a whole other set of enzymes because we've now synthesized saturated fats. We do need unsaturated fats for our health, and so there are enzymes to do that. Where does the synthetic process happen? I want you to focus more on the animal cell. And so what we do, most fatty acid synthesis takes place in the cytosol, okay, right here. And we will look at these other places like steroid synthesis and some other things in other compartments, but this is normally happening in the cytosol. All right. Here is the problem, though, with the synthesis of fat in the cytosol. Acetyl-CoA is made in the mitochondria. Okay? Remember, we take pyruvate dehydrogenase complex, and we take pyruvate and we react it with a whole bunch of enzymes and a whole bunch of cofactors, and we spit out acetyl-CoA. And that happens inside the mitochondria. Well, inside the mitochondria is not where we do the synthesis, so we got to get that out. Most of the time, acetyl-CoA is used to produce ATP and be broken down. Under certain conditions, the acetyl-CoA is exported. Okay, and so we need to get it out of the mitochondria. We do that by the citrate pyruvate shuttle. There is an ancillary benefit under certain conditions. We can also synthesize NADPH as part of this process. All right. So we get pyruvate inside the mitochondria. Pyruvate reacts with a pyruvate dehydrogenase complex. We made acetyl-CoA, and that's great. But if acetyl-CoA cannot enter into the citric acid cycle, which is its normal process where it's produced, then it builds up in concentration. Okay? Well, it'll actually get to citrate, but then citrate won't run any further. the citrate builds up in concentration. Usually citrate is a low in concentration, but when it gets high, it then can fit into the citrate shuttle, okay, and the transporter. Citrate makes it out into the cytosol. That's not where citrate is supposed to be. There's an enzyme called citrate lysase, the citrate is degraded into acetyl-CoA and oxaloacetate, which is the exact reverse of the citrate synthase. I now have my acetyl-CoA to go into creating fats. And we just did and highlighted this series of reactions. The oxaloacetate, at this moment in time, could go to synthesizing sugars if we needed it, but it can be reduced to malate. Malate can come back in, or malate can be reacted with NADP plus, and if we do have NADP plus around, we will do this reaction to create pyruvate, which is settled much better back into the mitochondria. And then the pyruvate can be carboxylated into oxaloacetate. This step right here, if you are remembering, this is also part of, or that reaction is similar to the reaction we see in gluconeogenesis. All right, how do we control all of this? When do we want to make fat, and when do we don't want to make fat? All right, we will only make fat if we have lots of energy, okay? And so hormonally, lots of energy and lots of starting material is signaled by insulin. If by chance you have glucagon, that means you're in starvation condition. This is not time to synthesize and use all your energy to make fat. If you've got glucagon going around, you're breaking down fat. All right, epinephrine is similar in terms of epinephrine. If you have that signaling, you're trying to optimize anything that will produce energy and turn off anything that makes energy. So insulin is going to get this roaming around. But insulin doesn't actually affect the enzyme. Okay, so let's go back and figure out what enzyme we're controlling. This one right here. Okay, acetyl-CoA carboxylase. Acetyl-CoA carboxylase makes malonyl-CoA, which is the committed step. Okay, if I control that, I don't do synthesis. If I turn it off, I don't have the starting material for synthesis. All right, and so inactive is this. I've got monomers of each of those proteins, and they're phosphorylated. Okay, that's in its inactive state. What does insulin do? I want to make it active. An active state is all the proteins create a polymer. There's nothing on it, no phosphate. So to polymerize, I have to take off the phosphate to get active to make synthesis of fats. Okay, so how does insulin control this? First of all, okay, insulin will come and upregulate something called protein phosphatase 2A. Okay? A protein phosphatase is the direct opposite of a what kind of protein? A kinase. Kinase puts on a phosphate, a phosphatase takes off a phosphate. And PP2A is a generalized phosphatase. So when insulin is released, it will make the phosphatase active, which cleaves all the phosphates off. When all the phosphates are off, it polymerizes, it becomes active. Okay? So that's how insulin affects the synthesis of fat. All right? We'll talk about what happens with citrate in a moment. Glucagon, on the other hand, okay, and epinephrine stimulate the pKa. pKa is what? Protein kinase A, okay? And that will phosphorylate. So glucagon gets a kinase active, phosphorylates them, and when they're phosphorylated, they dissociate, and when they dissociate and are phosphorylated, it turns it off. Okay? Epinephrine will do a similar upregulation of a kinase. So phosphorylated off, and individual, Dephosphorylated polymer on. Okay. Citrate. Now remember, to get the acetyl-CoA, I had to take citrate from the mitochondria and put it into the cytosol. That's not where it's supposed to be. Citrate concentrations go up. That's an indicator I've got lots of starting material for fat. So citrate's going to be a regulator. What kind of regulator does citrate behave like? Well, let's take a look. These are phosphorylated. They're individual. They're off. When citrate gets in here, they will bind and make them polymerize. Is this fully on? No, but it's partially on. How is citrate behaving? As a what kind of regulator? Okay, citrate's not a hormone, so it's not hormonal regulation. And hormones, from what we just saw, were covalent modifiers. They phosphorylated or they dephosphorylated. So it's not happening by a covalent regulation. It's an allosteric effector. And so it is a stimulant. It doesn't stimulate it 100%, but it does stimulate it. So you can have this situation in the body. Your hormones do not say synthesize, right? So that means insulin's not really high, but inside whatever cell you're in, you could have lots of citrate in the cytosol. The only thing that I want to do with citrate in the cytosol is actually make some fat, and so I can partially upregulate this and start the process, even in the absence of the hormonal signal. So it's an allosteric effector. All right. If I take a look, 16 carbons on a CoA. What is that? That is the ultimate outcome, right? That is the end product. What is the end product going to do? Well, the end product, okay, will bind to the inactive monomers, okay, or they will bind and make them monomers even when they don't have what? Phosphates. Okay, what kind of regulation is this? The end product coming back and stopping feedback inhibitor. If I have a buildup of the final product, don't go down that pathway, especially if I'm using so much energy. So the palmatic acid or the 16-carbon fat can make them stop. Okay? And so, in the absence of the end product and in the presence of citrate, this is the absolute highest level that you can have. But think about that. I'm in the cytosol, have lots of citrate that's not supposed to be there, and I have no fat. That indicates I have a lot of starting material, no product. Make that pathway go fast. Okay, and it is dephosphorylated, so it could be that the hormone is also saying, go ahead and go. All right. Now, AMP is also a control point on this. AMP represents inside the cell what? Not a lot of energy, right? If you got AMP, that means you've used a lot of ATP. Okay, so I want to tend to, you would think, if I have lots of AMP, stop synthesis. Well, let's see what happens. All right, this is a kinase, okay? It's going to seem to look like it's going to act on this polymer, and if by chance I take the kinase and I make it active, I'm going to phosphorylate everything and stop it, okay? Well, this kinase is AMP activated. So if AMP is around, no energy, it binds to this trimeric kinase. It will facilitate its own phosphorylation because AMP will upregulate another kinase, and it makes this one, this kinase, active, which then starts phosphorylating the active polymer and makes it inactive. So AMP upregulates a kinase to make sure that the enzymes will not make the malonyl-CoA, which then stops all of the synthesis. So that makes sense. So we've got lots of regulation here. Starting materials will regulate it, hormones will regulate it, end product regulates it, and energy charge. If I don't have a lot of energy, it's going to stop it. So we need to understand how all of these interplay. All right, I've synthesized them all. That's great. Well, we then need to do some unsaturation. We use that with molecular oxygen. It's a redox process, and we can make unsaturations, but there's a limitation. When I talk about fats, and if I put this delta 9, what does that mean? On the 9th carbon, that's where an unsaturation starts. Okay, I can have unsaturations at delta 2, delta 4, delta 9, delta 13. All right, here's the deal. Humans can only do unsaturations up to delta 9. All right, but we have lots of fats that need unsaturations past delta 9. Okay, arachidonic acid, which makes all the eicosanoids, we had unsaturations past delta 9. So that means something else has to do it. What is interesting enough is plants make the unsaturations. So if we can't make a fat, but we need that fat, what does it become? It's kind of like we need all 20 amino acids. We can't synthesize all 20 amino acids, so there are some amino acids that are called what? Essential. There are some essential fats, ones that we must eat. They are synthesized in plants. Okay? Do you think about it as plants being fat? Not normally, but have you ever had an avocado? Okay? Great source of natural fats. Okay? And you can get some essential fats in plant products, and you have to. And so for the later unsaturations, we have to get that from eating. The unsaturations that occur in terms of the reactions, ultimately we will use molecular oxygen, and we will use reactions from NADH or NADPH, very complex reactions with iron centers, but we will essentially, in the process, add a hydroxide, and then we will eliminate through that process. I won't ask you about that particular type of reaction. Other than that, it does require electrons and it does require oxygen.